So welcome to this video 4 of course G06 on transcriptomics and RNA-seq. We're looking at counting and normalizing depth in this part. So to follow on from our kind of outline for the library preparation and sequencing approaches for RNA-seq data, we can also consider the steps for our general pathway for bioinformatic analysis, though obviously it depends on what you're looking at to a large degree as well. So our first step always is FastQC to see did we actually um, did the sequencing work? Did we sequence what we wanted to sequence? Is there a lot of contaminants? So that's always our first step. We also include FastP is a very good tool for adapter removal and read trimming as well. There's options here that if we have a unknown species, which we're typically not dealing for modern day livestock genomes, we can use the likes of RNase-Phase or Trinity to do a de novo transcriptome assembly. If we do a very good reference genome, we can use either alignment-based quantification using Minimap2 and HT-seq. Optionally, that can also use TX import or Limma as well. And then we go for alignment-free quantification via Callisto and the sleuth pathway, or the Callisto to use TX import to then model that with Limma too. So there's different pathways here, and what we list here are only effective tools that are likely to be used in future years. So what we're thinking about when we're mapping reads to a reference genome is similar to what I've outlined before. That we have some reference sequence, transcriptome genome, and we're mapping our reads to that, and we can kind of estimate the coverage and depth based on the number of reads mapping to that particular location and assign that location properly. So if we're dealing with genome sequences and we're taking genomic DNA and mapping that to a reference genome, we just need to think about the location and the quality of that match compared to the relative qualities of the different potential matches across the different genome. For looking at RNA sequence, its different because it's more complicated because we have the different isoforms that are present. So we might have a red, green, and blue isoforms from the same different genes. We have different quantities of the different isoforms, and then overlaps between the different ones. So estimating the relative abundance is not necessarily straightforward. Also need to think about instances where you have different reads that can map to the same to any of these different isoforms as well. So they're not uniquely mapping. That makes it more complicated. So that means that when we think about our picture here, we have different isoforms 1, 2, and 3. We have different reads down here, some of which can be uniquely mapped, like these ones here, and some of which are more complicated. So this process is difficult, and that means, in essence, that we do a lot of mapping and remapping, which means efficiency is also worth considering as well. So that's called what are the isoforms and how much of them are there. So what we want to kind of think about is the minimum set of valid RNAs that are present, covering all possible fragments that could be there, even if they're just expressed at a single read. And ways of doing this include looking at the expectation and maximization algorithms to estimate the reads exon of origin and their expression levels, well, simultaneously, but also iteratively, that they use the information at the level of expression to estimate the reads exon of origin, because we'd expect a relatively even abundance of reads across a particular exon. So what we can think about is these mutually incompatible fragments where we have this yellow exon here, this blue exon here, and this red one down here that are all different. So blue and yellow and red are different, and if we map reads to them, we might find that they're different. So this is kind of shown in a kind of a de Boer graph, overlap graph layout. So what we can say is that they must have originated from different splice RNAs, because the yellow one lacks an exon, the blue one has a small exon, and the red has a big exon, and then the gray reads might be ones that are compatible with them all. So to think about the different paths that are present. So ways to address this is to assemble overlapping bundles of read alignments separately. So you kind of see that down here. We have those different paths for yellow, blue, and red here, where we've got a shared path going in, a shared path going out, and then these pair of bifurcations So we can kind of estimate the minimum number of transcripts that sufficiently explain the different paths that are present. Hence, you have this graph above where you have kind of no reads for this one, small clump here, and red ones that span that entirely. And a lot of the ideas behind this link from Dilbert's theorem that the number of mutually incompatible reads is the minimum number of transcripts needed to explain all reads, which is what we click on here. This is the minimum number for this kind of picture. And we're thinking about the largest set of reads where no two reads could have originated from the same isoforms. And we're going to think about the minimum number of paths covering all reads in the potential graph that we generate. So therefore, you can think about it, what are different reads and the different transcripts that we're doing a lot of back and forth, estimating the transcripts and their abundances and finding what the transcripts are, and then estimating the abundances. There's a lot of steps here, and that's why it's a kind of expectation maximization approach is one way of tackling it, because you have to repeat these things a lot. So that means we're estimating the probability of observing a read as a linear function of the possible different sources of RNAs that are present. So you can think of it this way in a diagram on the right, that we have black reads that can be across any transcript, and it doesn't really matter which one it is. There's no differences across them. And same with the ones down here, we can't infer which ones that the black reads come from. Whereas the red reads we know come from the ones down here. We might have blue reads that match the blue one, aligning here, and the same for the orange ones here. So the blue ones might map. We might have a kind of insert that spans a read or read pair here. So part of the approach of getting around this is called pseudo alignment. So this idea is simply to map the 5' end of the read only with imperfect matching. So the loss of accuracy is not an issue because we can do things faster, and that would mean that you can repeat this process many times and get a better overall estimate. So rather than just not doing the same level of compute, the compute is sent out is implemented differently to maximize the power from the data that you get. And essentially what we're dealing with, instead of mapping to k-mers, we're looking at k-mer classes, whereas not all k-mers might be represented if they belong to the same class. Instead, the k-mers are compressed into these classes. So if one pass is picked up, we don't necessarily need to map that read to all the k-mers associated with that particular group of k-mers. And pseudo alignment in essence means that we don't have to figure out every single letter in every word. Sure, it makes it easier to read, but if we kind of deduce what the likely ones are. So our typical units for describing this quantification process is TPM, transcripts per KB per million, or PKM and FPKM aren't used anymore because they're more biased approaches. Typically, this TPM is log scaled, so it's already a length normalized value, which is useful. And essentially, what we're doing is dividing all the recounts by the length of the gene in kilobases, and getting the reads per kilobase for each particular gene. And therefore, our transcript length is not a big of an issue. In terms of normalization, what you'll see most commonly for looking at gene expression analysis is that they use quantile or normalization, but other methods do exist. And a key assumption of this is that the mean transcript expression does not change in an experiment. There might be different inputs in terms of cDNA and so on at the start, but the average should be comparable across different experiments. So what might happen is that there's different correction approaches to essentially to make samples a little more comparable. So if you look at this graphically, and in terms of our picture here, we see this picture of different samples. Maybe there's different inputs, RNA levels. And so we get this pattern where differences between the green ones and the orange ones. And therefore we normalize that. What we're saying is that the medians shown here are the same. They have the same rank in all these distributions. And by correcting for the different ranks and using that as an index, we can ensure that the medians are the same in all these different experiments. And that should, in general, mean that the rank scaled values that we have here across all different experiments are comparable to each other. So this is locally expressed in this experiment, it's the same, has the same features as this one who's locally expressed in this experiment, and so on. So what we can think about is quantile normalization in practice. So you can imagine if we have these different samples here, A, B, C, D, E, F, and then we have our different genes that are present, A1, A2, A3, A4. For person A, these have different values. In person B, they have different values. C, we have different values, and some of these are tied, which is worth noting as well. B, D, and F, D, E, and F also different values. So what we can do first is get we can sort them by the different columns to extract the ranks. So 3, 7, 30, and 66. And you see for B, that's quite different. We've got 2 to 18 is the range. It's quite different from 3 to 36, 66. But the ranks are the same. So what we can do is take for all the top ranked genes, we can take the average of those, and that's the average expression level of all the top ranked genes. And equally for the bottom ranked here, got all the bottom ranked ones, and that's the bottom ranked gene expression for all the bottom ranked genes. And what we're going to do then is replace this value 20 with each of the bottom ranked genes there. And that's our normalized value for all the bottom ranked genes. So we put that in there for these ones. You see it's 2.2, 4.2, 11, and 20 for all of them, except for C because that is ties. So we take the average between ranks 1 and 2 and 3 and 4 for each of those. And then we have these samples. We essentially unsort them into the order that they were at the top. So this moves 3, 30, 66, 7. So this will become, what's it going to be? 2.2, 11, 20, and 4.2, I think. Let's see. Yeah, 2.2, 11, 20, and 4.2. So you see down there for C, it was 9, 9, and 3, 3. Now it's 15, 15, 3.2, 3.2. So now this is a normalized matrix of different data. This quantile normalization procedure has meant that we can compare these different samples to each other and see that the actual values we examine in gene expression experiments are not the original values because they're skewed by the library preparation approaches and the application process during sequencing as well. There are also other normalization approaches, many diverse ones that are not outlined here. One that's common in certain RNA-seq tools is called the trim tail submedians. That's another scaling approach. So we have to consider then that it doesn't quite use ranks in this instance. So if we consider that we have experiment A where most of the genes have a low expression values, we have many zero values, and experiment B is a normal spread of values, we scale it by the library size. So B is more reads, therefore we can scale it by the number of reads that are present. So for the median of A is 4 and the median of B is 14. Then we're just going to divide the gene A by the library size of A and gene B by the library size of A, and then I take the log 2 of that to compare A versus B for that particular gene G. That's in essence what we're doing. It's a different approach, not using the ranks, which is more accurate. It really depends on your data that's of interest. They're just different approaches to solving the same problem. And therefore you'll see that different tools will use slightly different normalization approaches. So it's worth being aware of that. So please see in our next video that before the part we have to consider RNA-seq file types.